Welcome back to our discussion of the regulation, neuroendocrine regulation. This is Chapter 8, the regulation of hormone levels in the bloodstream. So Chapter 7 helped us to understand some of the bases for in our brains visualizing what the plasma concentration might be at any one point in time. And now let's take a little more consideration of the mechanisms regulating those hormone levels. So we are keeping in mind the bathtub approach, water in versus water out. We have to synthesize our water and secrete our water, right? And then we transport through the bathtub, having the effects either, remember it's synthesis, secretion, regulation, and function that we always think about. So regulation is what we're thinking about here, and we touched a bit on function before. What we have to do first is to remind ourselves that the concentration, let's just say the effect of a ligand on its target cell depends upon the concentration of the ligand, hormone in our case here, and the receptors on or in the cell. And so what we're talking about here is regulating the concentration of the ligand within the plasma, because that's what we're sampling to measure the levels. And if we remove something like this, the kidneys is in effect removing or clearing luteinizing hormone, then we are removing it from the interstitial fluid through mass action throughout the body, and decreasing the concentration of ligand in the interstitial fluid at the presence of the target cell, potential target cell. And then the target cell takes care of the concentration of the receptor through its own mechanisms. So that's what we're trying to regulate, is the effect on the target cells by regulating the level within the interstitial fluid, essentially the plasma. And we can convert a hormone from active to inactive state, or just remove it from the plasma in its active state, as in the case of the LH here. So this is what you want to be picturing in your mind, this entire system, our bathtub, and how all the compartments, the fluid compartments, plasma and interstitial as the extracellular fluid, are experiencing the flux or the fluctuating, let me say, concentrations of ligands. One quick note that Chapter 8.1, measuring hormone levels, will be covered in the methods lectures, so we're not going to talk about it here. So we're going to begin with 8.2 and kind of skim through the rest, the mechanisms regulating hormone levels and things of that nature. So we go first to figure 8.2. So this is a comparison of serum and urinary LH levels in 3-hour periods near the time of ovulation in a human female. So 0 hours was defined as the time when there was a 2-fold increase in urinary LH concentration above baseline levels. From back here, you see you go roughly from 50 to 100 or more, right, at the time 0. And it looks like about 125. The assessments of serum LH are related to this point. So this is redrawn from Karen et al. In 1980. Okay, so let's look at our axes. Whenever we see a graph, we go to axes first. The scale is time, and the units are hours. So 0, plus 6, 12, 18, 24, 28, and 32. So it's a it isn't a 4-hour interval. 6, 6, 6, 6, and then 4, and then 4, right? So these shouldn't be uniformly spaced here. Okay, and then minus 6, 12, 18, 24, and 30. So it's equal 6 going in that direction. So what troubles me here is where they have ovulation so close to the serum LH. But anyway, let's get back. That's our x-axis. Scale is time, units are hours. Here, scale range is 0 to 200. The urine concentration in international units per liter. And for the plasma, the serum, on the right-hand side, the scale is serum concentration in international units per liter as well, and from 0 to 90. So they use different scales to kind of have the lines overlap a bit so that we can see when the peaks might correspond or not, right? And what we would think is that the urine would trail the plasma a bit, but oddly enough, so there might be some concentrating effect, I guess. But then here, the plasma is more concentrated than the urine. No, excuse me, it isn't. That's right. We would have to, I made the mistake of putting them both on the same scale. So the plasma drop from here is about 65 down to about 60 international units per liter, and kind of over that same time interval, the urine is going from 150 to about 110, right? So the magnitude of drop is much greater in the urine over this same time period than the drop in the plasma. Okay, so what are the dynamics contributing to those differential concentration drops? Maybe no one really knows. The accuracy of the tests might not be so perfect, and then collecting in humans, how much water did they drink in between and stuff like that. So was the osmolarity of the urine constant throughout here, right? So if the urine became more dilute, you might have a bigger drop, right? Plasma should be, osmolarity should stay about the same. Okay, the urine osmolarity changes to maintain plasma osmolarity. So that's one possible confounding factor to explain why we get a 40 international unit per liter drop in the urine versus 65 in the plasma. Okay, so that helps us to understand that part of it. But the important thing is that both peak before ovulation, and my recollection is that it's about a day and a half before ovulation that you get the plasma peak in LH. So in my opinion, this arrow should be shifted to the right, even off this scale. So I don't, this is baffling me, obviously, because when I talk about the menstrual cycle, we talk about the LH surge in the plasma coming about a day and a half before ovulation, and they're having it just a matter of hours here. So again, this makes us turn to the literature, review articles. We're questioning our textbook that's citing primary literature in the 80s, and so here 20, 30 years later, do we have any data to improve upon this? Again, this is in human females. We don't know what age they are, but we would assume they're in their mid-reproductive age, later 20s, I guess that would be, right? Current culture, generations ago, it might have been late teens, right? Anyway, we also know that over a period of time, we have hormonal feedback mechanisms. What we'll find in the study of the menstrual cycle is that estrogen levels are also increasing so that's hormone 3 increasing as hormone 2 increases. And what we'll find is that in females during the reproductive cycle, there's a switch from negative feedback over here to positive feedback, so that estrogen causes this LH surge. That's what we want to start thinking about, is the feedback. So remember, we said we're talking about stimulus, secretion, regulation, and function. So previously we kind of considered function, cyclic AMP in various target cells, target tissues. And so now what we're thinking about is regulation. Okay, so you still have target cells, you got source cells, source of the hormones. Is it hormone 3 on hormone 2, hormone 3 on hormone 1, or should be hormone 2 on gland 1, hormone 3 on gland 2, what is it? So that's the game we're playing here. So the feedback mechanisms, and they can be positive or negative feedback. So feedback is just a type of regulation. Feed forward is hormone 1 stimulating hormone 2 stimulating hormone 3. Now we're talking about feedback, and it can be positive or negative. And we saw in females it's negative for one set of phases in the menstrual cycle, thinking about humans, and then just before ovulation, pre-ovulatory phase of the menstrual cycle, it'll switch to positive feedback. So the same hormones and target cells, and there's just a switch in the molecular machinery to go from a negative feedback to a positive feedback. Okay, so feedback can be both negative and positive, as we know. Excuse me. However, in general here, we know we're thinking of negative feedback for regulation. So one endocrine gland, one hormone is the idea. The autocrine self-regulation for the first order loops, and the neurohypophyseal hormones are the ones we think about for that. The second order feedback loops, 2 gland, 2 hormone system, where the hypophysiotrophic releasing and inhibiting hormones would be involved. In the third order feedback loops, the 3 gland, 3 hormone system comes into play, where you have 3 levels of feedback: short, long, and ultra short. Ultra short is first order, it's like that, but it includes hormones 2 and glands 2 and the hormones. Okay. So these are definitions I extracted from the textbook, and probably the thing for you to do if you're interested in getting a more thorough understanding of this and the current concepts of these feedback loops is to take a look at our online texts, endotext and things of that sort, or probably won't find it in review literature anymore. It'll be in the textbooks. Anyway, this is what we're thinking about. 3 gland, 3 hormone system. So hypothalamus is gland 1, anterpituitary is gland 2, peripheral endocrine glands are glands 3. The hormones 1 are releasing and inhibitory hormones. Hormones 2 are the protein polypeptide, large polypeptide hormones. And hormones 3 are typically steroids, the lipophilic hormones, or thyroid hormones. And then the effector cells. Now there are some peptide hormones 3, if we think of growth hormone stimulating the release of insulin-like growth factor, IGFs, from the liver, and things like that. But anyway, we typically go lipophilic hormones, hydrophilic hormones. So the ultra short loop is hormone 1 back on the hypothalamus, if you want to think of it that way. Short loop is hormone 2 onto gland 1. And then the long loop would be hormones 3 onto gland 2 and gland 1. This makes us ask questions about the model for homeostatic feedback mechanisms. And in this case, I challenge you to determine whether this is a negative or positive feedback. So think of it, we have a disturbance, a change in the level of a hormone, a controlled system that has an output. This is stimulus, this is release. Okay. Then we have a sensor that has a certain set point with a comparator. Did we go above the set point or below the set point? Okay. So disturbance, you remove glands 3 with an oophorectomy or a orchiectomy is the males, and Therefore, the controlled system will have a tremendous decrease in the output that'll fall below the set point for the sensor that performed that comparator function, and then we have an inverse amplifier to give the feedback. Okay. Now let's think of one where it would go up. So we would inject testosterone into a male or a female, and so in effect, controlled system gland 3 would have an increased output of hormone 3. The sensor comparator determines that that is above the set point. Say it's 10 above the set point. Then you inverse amplify that to a negative 10. You give that feedback to the controlled system, pituitary gland, and it's going to decrease by 10. We converted a plus 10 here to a negative 10 to come back and decrease hormones 2, which would be LH and FSH, to decrease endogenous testosterone, so that we would end up with only the exogenous testosterone that we put into the system to get us back to our set point. Does that make sense? So if you add exogenous hormones 3, this is our controlled system, our 3-gland 3-hormone system. So we have added hormone 3. The output of hormone 3 at some sensor determines that hormone 3 is now above exogenous plus endogenous, 10 above the set point. Turn that into a negative 10, and then we dampen the system at the anterior pituitary so that endogenous testosterone synthesis and secretion is brought down, so that plus the exogenous brings us back to the set point. Make sense? Now think of the 8.5 as from the textbook as prolactin levels increase in the circulation. What effect do they have on dopamine release in the median eminence? So now we're talking about constitutive release of prolactin here. Maybe there's a prolactin releasing factor. Dopamine is inhibitory. So if prolactin goes up by 10, and the sensor at the set point says it's above, we have a comparator, turns it to a negative 10, and then dopamine is going to inhibit this. Which direction will dopamine go? And is this negative or positive feedback? I won't answer the question. You have to think it through yourself, and I assure you that you'll see this on the test. Okay, so that's the way that I want you to be able to problem solve and critically think. Let's go back. So we have gas pedals and brakes, and comparators that turn excesses or absences into the inverse value to drive the gas pedal or brakes accordingly to adjust the system back to our set point. That's homeostasis, right? So, illustrate this and think about prolactin going up, and what do you need to do to dopamine to bring it back down if dopamine is an inhibitor here. And so does this really fit as a negative or a positive feedback, and how would you justify your argument? Okay, now let's think of neurotransmitter and neuropeptide control of hormone levels. So it's kind of appropriate with the dopamine, although the dopamine is not really a neurotransmitter. It's acting as a hormone, a neurohormone, because it's synthesized and secreted by dopamine neurons in the hypothalamus into the pituitary portal plasma, and then acting in an endocrine fashion at the remote, still pretty close by, but it's remote compared to a synapse where a neurotransmitter would have its effect. Anyway, we kind of have all that in mind of autocrine, paracrine, and endocrine, and we just keep it in mind for the dopamine on the lactotrope in the previous example. Thought question. Now we're just thinking of neurotransmitters at the synapses, the neuroendocrine transducing mechanism in the hypothalamus, or maybe neuropeptides as well. Okay, so here we're thinking, and this is a 1999 J Neuroscience article, and anytime you see an article in here, typically the article is available, it's required reading, posted on your online course site. So the hypothalamic integration of stimuli influence neuroendocrine release. This is actually from an article that I published on the distribution of serotonin axons in the field mouse, and there was a silver stain on the antibodies that bound to serotonin in their axons throughout the hypothalamus, and then used a dark field illumination to make the axons turn white and the background turn black. It's just an easier way to see their distribution. So in this field mouse, a lot of serotonin axons coming up through the medial forebrain bundle and the lateral hypothalamus over here. And here's the fornix, and you can see the areas of high terminal density and low terminal density, or it may just be that you've got the big neurons here in the magnocellular portion of the paraventricular nucleus. Terminal density is lower simply because you've got big plump neurons. Anyway, the ventromedial hypothalamus has a lower density of terminals and axons, quite obviously. Anyway, this just shows us that there's a lot of axonal input. These are just serotonin, so think of norepi and then all the peptides. So there's a lot more axons. It doesn't seem like there's room to fit them in, but somehow they are all there. There might be colocalization, but not too much with the serotonin. So this reminds us of table 8.2, input to the endocrine hypothalamus with neural inputs, sense organs like vision, smell, taste, hearing, and touch. Think of stressors. Animals that can smell predators are going to turn on their hypothalamic pituitary adrenal axis. You taste something foul. You can always think about any of these could be aversive or rewarding and bring about changes in some endocrine input. Exteroreceptors such as suckling, coitus, pain, and temperature. Interoreceptors inside the body, uterus, cervix, blood volume. Chemoreceptors. And chemoreceptors includes the, along with the volume, the concentration of the blood, right? Antidiuretic hormone, sensing, the ADH neurons of the supraoptic nucleus in the hypothalamus, sensing the osmolarity of the blood, right, which reflects blood volume, and it's also kind of a chemoreceptor. Okay, then the stress, the physical, emotional, or overcrowding, time-dependent rhythms and sleep-wake cycles, cerebrospinal fluid inputs such as hormones, ions, other substances such as peptides, and then circulating system inputs such as hormones, gonadal and adrenal steroids, lipid-soluble, so they go right through the blood-brain barrier. Again, we're thinking about the endocrine hypothalamus. Thyroid hormones, the same. Pituitary and hypothalamic peptides can leak through at a very low level, but still, this is how they're contributing to the hypothalamic peptides getting back into the hypothalamus for the ultra-short loop negative feedback. And melatonin. The neuropeptides, substance P, bombesin, and several others through circulatory systems. Again, somehow getting into the hypothalamic interstitial fluid space and binding to receptors on the neurohormone, neuroendocrine neurons, and in these cases probably modulating their activity. Other substances such as glucose, amino acids, and blood gases, like amino acids, are pretty potent stimuli for the growth hormone axis, so growth hormone releasing hormone having to be released to increase growth hormone secretion and plasma levels in response to certain amino acids. And then other properties of blood such as temperature and osmotic pressure, as I said up here. So the source is given in this table. So this then makes us think of temporal scale. So I've tried to emphasize that all along. Anytime we see a graph, look at our axes. What's the scale? It's a time scale. What's the units, duration of hours? And so we think of our temporal resolution and what would be the biologically accurate temporal sequence of events, right? Hormones 1 should change and peak and then drop back down before hormones 2 in the pituitary and before hormones 3, and the response to hormones 3 in the target tissues, in this case muscle, to cortisol should have a progressive wave. I always call these a bell-shaped effect, but this is not really bell-shaped. It tapers more slowly. Remember, in this case, hormones 3 is a steroid, lipid-soluble. Therefore, it has genomic effects on its receptor-mediated mechanisms in the target cell. So it depends upon gene expression changes to have this increase, and then a reversal of those gene expression changes all the way out into the protein and its activity and the life cycle of the protein out in the muscle cells to reverse that effect. Okay, so we see this rise phase and falling phase. They have different temporal scales for each of these hormone profiles, and that has to do with the secretion rate, our bathtub again, the rate of water going in, secretion with synthesis, rate, and the removal rate, the clearance. Okay? But we should be able to get a reasonable temporal delay between peaks 1 and 2, 2 and 3, and then so this is the stimulus response part of it, right? And regulation for our feed-forward system being temporally delayed between peaks, and then our function, our stimulus response, regulation, and function. And part of the turning off, there's some, this rise phase should contribute to this fall phase. This rise phase of hormone 3 should contribute to this falling phase here, as well as clearance. Oops, sorry. So keep that in mind, right? So we have regulation, feed forward regulation driving the rise in the peak, feedback regulation contributing to the fall, but also clearance, the mechanisms for clearance, which may include decreasing synthesis and secretion, right? But typically we think of deactivation and removal from the tissue fluid or plasma that we're tracking here or imagining. Okay, and then we have to think about the function in the target organ. What factors contribute to the rising phase, the magnitude of the peak, the duration of the peak, does it plateau, what triggers the fall, and then the rate of the fall back to homeostatic levels, right? And how does that contribute to the temporal profile of the overall response? Right, so a dinosaur chases you. You want an immediate response to get the cortisol into the blood within a matter of minutes back here, but then you have a couple of hours of effect on the muscle, right? Because you run away, burn up the glucose available in your blood, and then you want the glucocorticoid contributing over a longer period of time to keep giving glucose into the blood to replace what you burn. Because you're not going to stop and eat, because you'll get eaten by the dinosaur. So we can kind of make sense of it that way. But most importantly, we think of this temporal scale and what contributes to the rising, plateau, amplitude, plateau, and falling phase, and overall duration from origin to peak, and the duration from peak to return to baseline levels. Okay, so now let's take a look at an example, figure 8.10, moment-to-moment fluctuations in circulating LH concentrations, again LH, but in a castrated male rhesus monkey. So hormones 3 are removed, testosterone, and in panel A, we have 1 day before, panel B, 4 days after, and panel C, 8 days after the implantation of a silastic capsule containing testosterone, which restored the level of this steroid to the normal physiological range. So in males there is some circadian rhythmicity, but this type of silastic capsule, once it balances out, there'll be a little burst at first when it first starts to release its contents, then it'll be stabilized. You won't get a circadian rhythm, but you'll have a constant plasma level generated within the normal physiological range. So depending upon the release rate from the capsule and what concentration of testosterone you put into the capsule, and the clearance rate, once you let it stabilize, reach some type of artifactual homeostatic status, right? Then you can begin your study on the animal. And here they did 1 day before, 4 days after. So they gave it 4 days to kind of settle into place, and then 8 days after. So hormones 3 would be restored to physiological range levels. And those are just measured. And then the arrows indicate the LH pulses. Note the retardation of LH pulse frequency and presumably, therefore, the inhibition of the hypothalamic GnRH pulse generator 4 and 8 days after initiation of the testosterone replacement. So from the discussion of Chapter 7, we tried to picture the stimulating ligand, the second messenger systems, and events in the target cells that would bring about calcium waves, so oscillations are what we ended up reaching. And we're envisioning this in both the GnRH neurons of gland 1 for the HPG axis and the gonadotropes, in this case the one for LH, in the anterior pituitary. So in each of those cells for hormone 1 and hormone 2, we're trying to picture what would generate pulsatility in each of them. So in gland 1, we're talking about rhythmicity of action potentials, because we have action potentials coupled to the exocytotic release of GnRH into the portal vessel system. And then those pulsatile waves of GnRH reach the gonadotropes in the pituitary and will contribute to calcium oscillations for, the cyclic AMP is going to maintain the synthesis going to the nucleus. So we coordinate synthesis and secretion in the anterior pituitary such that we can sustain the pulsatility. So somehow the 1 day before, so this is the orchiectomized, ORCIECOTMIZED, orchiectomized, gonadectomized male, right? The male gonads are called orchids. They are hidden away, I guess, and descend into the scrotum. So somehow they come up with orchids, I guess, for that. Anyway, so the orchiectomy is the male gonadectomy, or castrated, right? We just say castrated male. So let's take a look. Time is our scale. Units are hours. So 7, 8, I guess it's military time a day. 700, 800, 900, 1100, 1300, 1500, and 1700, right? So noon is right here in the middle. So midday is right here in the middle. And our y-axis, we have 3 windows, and each is plasma LH in nanograms per ml. And we've got 40, 80, and 100, and 20, going up to probably about 300, I guess, there, and about 300 for day 4. And then day 8, we don't even have to go up to 80 because plasma levels are so low when the testosterone has had its full negative feedback effect. So we see the pulsatility coming through. So this is 1 hour. So we've got 1, 2, 3. Looks like every 20 minutes maybe they measured. So here 1, 2, 3, 4, 5, 6. Every 10 minutes, I guess, right? So every 10 minutes. So here's 10, 20, 30. Forward, and then down, and 10, 20, pretty much 30. So it's about a 30-minute cycle, right? When there's no testosterone. Excuse me. when there's no testosterone present, we got about a 30-minute cycle, right? And then 4 days after the implant, we switch to what looks like 1, 2, 1, 2, 3, 1, 2, about a 2 to 3 hour cycle. And then 8 days after the implant, there's really no cycling, ultradian cycle, right? Less than a day cycle. So these would be ultradian cycles. So again, hormones 3 are lipophilic, therefore they have genomic effects on their target cells. So what we're seeing is a gene expression change in neurons, GnRH neurons of gland 1, and the luteotropes, if you want to call them that, the luteotrope subpopulation of the gonadotropes in the anterior pituitary. Collectively, the two of those cells, endocrine cells, have changes in gene expression that first reduce the pulsatility, and look, it also brings it lower at the trough. We have a peak and a trough, right? So the peak and the trough. So the trough is brought to lower levels, below 40, without any testosterone present. It barely dips down. If you averaged it, it'd be somewhere around 75, maybe would be the average trough. And the average peak is somewhere around maybe 100, 110. And then you can see that we consistently get up to 120, but the amplitude is exaggerated because the trough is depressed down to below 40. And then finally, the rhythmicity is attenuated almost completely. There's little pulses, I guess they're still showing the arrows here for slight pulses, but the amplitude of the pulse and the rate of return to baseline is altered again. So we've got 1, 2, 3, almost a 3 hour. So we shift from a 1, 2, 2.5, 2 to 3 hour down to 3 hour intervals between our peaks, and the amplitude, rate of increase, rate of decrease, all that is changing. So the whole dynamics of stimulus, synthesis, secretion, biological activity, and deactivation and elimination, clearance, are all changed based upon changes in gene expression of the testosterone on the GnRH neuron and the gonadotrope in the anterior pituitary. Now, the reason that I'm stating that there can be an effect of testosterone on the GnRH neurons is that there's recent evidence here in 2013 for a subtype of the estrogen receptor. Testosterone be converted to estrogen and then bind to the estrogen receptor and have negative feedback both in the anterior pituitary and into the GnRH neurons. So you'll see in other lectures that I recorded before 2013 that nobody believed that there could be a negative effect of hormones 3 of the HPG axis on gland 1, the GnRH neurons, because nobody could detect estrogen receptors there or androgen receptors. Well, it turns out that there's one subtype of the estrogen receptors that can be detected there, so now the way of thinking is a little bit different, right? As techniques develop, we learn more as we proceed, and it impacts our theoretical framework. Okay, anyway, we wrap this up with the review questions responsible for on the test. What is the effect of removing testosterone on the secretion of LHRH and LH? And would you expect to find estrogen receptors in the pituitary gland? I guess you would. I just answered it. Well, why? And how? Okay, so review the lecture in Chapter 7, and then the subsequent one we'll talk about steroid synthesis, and we will eventually answer these questions, but you should be seeking the answers on your own. So that completes our presentation.